Hey guys, welcome to AP Biology, and we're going to talk about the light reactions of photosynthesis. So this is follow up to the lecture Photosynthesis Part 1, which was just a brief overview of photosynthesis. Now we are going to concentrate on the light reaction. So I like this little diagram because it shows everything you need to know basically. We see light is providing the energy. And what's that going to do? We're going to have water coming in as a reactant. We're going to have oxygen coming out as a product. We're also going to produce NADPH and ATP. So let's go ahead and do our deep dive into the light reactions. So first of all, here's this introductory diagram that we saw earlier. And of course, this is occurring in the chloroplast. And once again, we see water coming in. It's going to be happening. All the reactions are going to happen on the inner membrane, the thylakoid, okay, where light is going to be absorbed by chlorophyll. Water is going to be split by light, and oxygen is going to be released from the water. And using a combination of proteins in the electron transport chain, we're going to make ATP and NADPH. Okay, so just a quick reminder once again, chlorophyll is going to be the molecule that is going to absorb the sun's energy, and it's going to be magnesium atom in the center of the chlorophyll molecule. And if you remember, magnesium has two valence electrons, and those two valence electrons start off at a low energy state. And when they absorb the energy in the photon, they are going to get boosted to a higher energy state, i.e., those electrons are going to move to a higher energy shell that's typically further away from the atomic nucleus where the protons and neutrons are located. So that is the conversion, and that's the first step is the conversion of that light energy into a form of chemical energy, and that is in the form of high energy electrons. Okay, so that is those high energy electrons represent a form of potential energy. And so all of this is again occurring in the chlorophyll molecules, and the chlorophyll molecules are just not spread out randomly in on the inner part of the chloroplast, i.e., the thylakoid membrane, except what they are done is that they are organized into these clusters of chlorophyll molecules that we call a photosystem. And if you remember from earlier, there are two photosystems. There's photosystem 1 and photosystem 2. Okay, so the light dependent reaction takes place in three main stages. The primary photo event, which is the absorption of photons by the magnesium electrons. Then electron transport, which is going to be those electrons once they leave the magnesium atom at the center of the chlorophyll molecule, they're going to move down the electron transport chain, and in that process, they are going to pump protons against a concentration gradient. So we're going to use the energy of those high energy electrons to pump protons and create and maintain a concentration gradient. And then we're going to use the concentration gradient to make ATP via the process called chemiosmosis. So that's all the stuff that is happening in the light dependent reaction. So let's just look at this particular diagram and basically what happens. Here we see a photon of light coming down, striking a chlorophyll molecule. Well, those chlorophyll molecules don't immediately transfer those high energy electrons to the electron transport chain, except what does happen is that the energy is transferred to adjacent chlorophyll molecules, and it kind of is bouncing all around. And think about this, there's literally millions of photons striking all these simultaneously, and this energy is being transferred from chlorophyll molecule to chlorophyll molecule until it randomly hits the what we call the reaction center chlorophyll. It's a special chlorophyll molecule, and those electrons now are going to get boosted to a higher energy level, and they're going to get transferred to a protein that is the first member or first protein of the electron transport chain, and that's what we call the primary electron acceptor. Okay, so the energy has been transferred from a photon to the electrons of magnesium, and those magnesium electrons then get transferred to another protein that is the number one protein in the start of the electron transport chain. Okay, and what we're going to look at first is something we call non-cyclic electron flow. Why is it called non-cyclic electron flow? Well, let's just follow some electrons and figure out why. So, again, we have energy coming in the form of light. And I want you to look at this diagram first of all. We see photosystem 2, photosystem 1 here. We see over on the left-hand side, we see the energy of the electrons. So this represents down here low energy electrons. And as we go to the top part, high energy electrons. So number one event is that we have light coming in. And that light's going to do two things. It's going to energize electrons, and it's also going to be used to split water. And when water is split, it's going to produce oxygen, two protons, and two electrons. And these two electrons are important because remember, magnesium at the center of the chlorophyll molecule has two valence electrons. When those electrons get boosted to a higher energy state, they're going to be transferred to a protein in the electron transport chain. That's called the primary acceptor. Well, now magnesium is now missing two electrons. It needs to get those electrons. Where are they going to come from? They're coming from water. So water, those two electrons from water are going to fill that electron hole in this primary or in this reaction center chlorophyll molecule. Well, now let's look. We have these two high energy electrons up here. They're going to move down the electron transport chain. And I say down, they're moving down in terms of energy. Because this is high energy, this is going to be low energy down here. And what are we doing with the energy of those electrons? We're going to use them to pump protons against a concentration gradient. Okay? And these little PQ and PCs, those are just different proteins. They're mobile proteins that can move in the phospholipid bilayer of the thylakoid membrane. They're going to be moving those electrons from place to place. So after those electrons have used their energy to pump protons against a concentration gradient, they no longer have much energy. Where are they going to go? They go into the electron hole in the photosystem 1 chlorophyll molecule. Okay, because they're going to go into this hole, they're going to get boosted up to a higher energy level by light incoming. They go once again to a primary electron acceptor, there's my two electrons, into a much shorter electron transport chain. And in this case, this electron transport chain, the electrons are going to be used to reduce NADP plus into NADPH. Okay, so that's where the electrons go. They go to NADPH. And it's called non-cyclic because the electrons begin in water and they're going to end up here in NADPH. And we're going to use that eventually in the next stage of photosynthesis, the light-independent reactions or the Calvin cycle. So that is non-cyclic electron flow. The electrons move from water into photosystem 2 and then down through the electron transport chain into photosystem 1. Back on up, they get boosted back up into a higher energy state, go down a short electron transport chain where they are going to be used to reduce NADP to NADPH. And notice that this is being carried out by an enzyme called NADP reductase. Cyclic electron flow, on the other hand, the only thing that cyclic electron flow does is it's going to make ATP. Okay, it's going to make ATP, and the electrons are flowing in a cycle, in a circle. They're going from photosystem 1, they get boosted up into a primary acceptor, and instead of going down to NADP reductase, they're coming back into this cytochrome complex. What is the cytochrome complex, you ask? It is it is a proton pump where that utilizes the energy in electrons to pump protons against a concentration gradient. And we'll get to what happens with that concentration gradient a little bit later. Okay, So where are these photosystems located? Again, they are located on the inner thylakoid membrane. All right, and remember the inner thylakoid membrane is highly folded. What does that do? That allows for increased surface area, which allows for more photosystems, and it also allows for more electron transport chains, and also the ultimate molecule that we need here, which is ATP synthase. Okay, so here's another view of that electron transport chain, a more realistic view of that electron transport chain, where we see photosystem 2, which is a protein complex embedded in a phospholipid bilayer of the thylakoid membrane. Plastoquinone, PQ, is a mobile protein element that can transport electrons from photosystem 2 into the cytochrome complex. The cytochrome complex is a multi-protein proton pump. Multi-protein proton pump. Wow. Okay, that is going to pump protons from a low concentration of protons in the stroma into a high concentration of protons on the thylakoid space. This is a form of active transport. No ATP is being used in this. However, we're using the energy of these high energy electrons. And if you remember, after they go through the cytochrome complex, those electrons have lost their energy. They now go to another mobile protein element of the phospholipid bilayer called plastocyanin. Okay, and plastocyanin transports those electrons to photosystem 1, where once again they're boosted up to a higher energy state by light, and going to another mobile protein element called ferrodoxin, which then brings those electrons to the big protein complex called NADP reductase, which now is going to take NADP plus some protons plus the electrons from the electron transport chain in order to reduce NADP plus into NADPH. And we've now have followed the electrons through the electron transport chain. I also want you to notice that what have we done in that process? We've increased the proton concentration inside the thylakoid space. It is about 1,000 times more concentrated of protons inside the thylakoid space than it is in the stroma. And that is creating lots of potential energy. And again, potential energy is the ability to do work. And we're going to use those protons to make ATP as those protons diffuse down through another enzyme complex, protein complex called ATP synthase. So we've used the energy of these electrons to pump protons to create a concentration gradient, and now we're going to use this concentration gradient to make ATP. This coupling is called chemiosmosis, which we'll come across again in cell respiration. Very similar process in cell respiration, okay? Except in cell respiration, the energy is coming from complex carbohydrates and fats and proteins, whereas in photosynthesis, the energy is coming from light. So let's look at another picture of these photosystems. So here is the thylakoid membrane, and here we see all of the various thylakoid discs. And the chlorophyll are the photosystems, or chlorophyll, are these little green things. Notice that the little green things can be scattered anywhere kind of within this thylakoid membrane. All right. Whereas ATP synthase, remember what has to happen with ATP synthase. We have to diffuse high proton concentration, or diffuse protons from a high concentration to a low concentration in the stroma. So the ATP synthase must have access to the stroma. So that's the reason why there's no ATP synthase right here, because there's no access to the stroma, because these thylakoids are stacked on top of each other. Okay, so that's just showing you how this is organized within the chloroplast. And again, we already mentioned this, the increased folding is a result of natural selection that has evolved into the complex chloroplast that we now know today. And if you remember endosymbiosis, this original inner chloroplast, this original thylakoid membrane, was probably just a round bacterium shaped organism, but over time it started to fold and increase the surface area and make it more efficient, and natural selection favored those individuals with more folding, more efficient photosynthesis production. Okay, so again, the role of water in photosynthesis is to supply the electrons needed by the electron transport chain in order to fill that hole that is left when magnesium loses those two valence electrons. So, water is supplying the electrons. A byproduct of that water, of course, of that splitting of water, is going to be oxygen gas, which is really important for us because without it, we would not be here. Complex multicellular life would not be here. There would be no life on land because oxygen is necessary to make ozone, which is O3. And ozone, of course, protects us from the highly DNA damaging ultraviolet radiation. So, ozone in the high level atmosphere is extremely important for protecting us on life on land. However, ozone at ground level, which is a result of fossil fuel burning, is highly destructive to living biological tissues. So, it's like one of these two edged swords. Anyways, that's where water plays its role in photosystems 1 and 2. And again, photosystems do collect the energy of sunlight, and we're going to look at another picture of it. So let's look at another model. There is photosystem 2, okay? And photosystem 2, oftentimes called P680. Again, there's that light being bounced around, the light's coming in, and it's energizing these two electrons in the magnesium's valence electron shell. Okay, they get boosted to a higher energy state. Those two electrons go to a primary protein acceptor. We're going to split water in that process. Those electrons from water are going to be used to fill that electron hole in the reaction center chlorophyll. And then those high energy electrons are going to move down the electron transport chain where they're going to be used, where the cytochrome complex is going to use the energy in those electrons to pump protons in order to create a concentration gradient and use that concentration gradient of protons to eventually make ATP through ATP synthase. Those electrons, after being depleted of their energy, go to photosystem 1, where once again they are going to be energized, move up to another primary acceptor, go down another short electron transport chain where those electrons are going to be used to reduce NADP to NADPH. I do want you to know that NADPH and ATP are both going to be made in the stroma because the protons diffuse down through the ATP synthase, which is going to have its ATP anabolism site on the stroma side, and the same thing with NADP reductase. It's on the stroma side of the thylakoid. Okay, and that little arrow that just popped up, that is just showing the direction in which non-cyclic electron flow cyclic electron flow would move. Okay, from photosystem 1 to the cytochrome complex and then back in and just go in a cycle pattern there. Remember, these are all redox reactions where a compound A in this case is going to be oxidized. Okay, right here, compound A is a reducing agent. Why? Because it's going to supply the electrons to reduce compound B. Compound B is going to be an oxidizing agent. Why? Because it's going to allow for um, uh, compound A to be oxidized. How's it doing that? Because it's going to pull the electrons off of A. Okay, and so A is going to be reduced, or excuse me, B is going to be reduced, and A is going to be oxidized. Okay, A is oxidized, it's losing electrons. B is reduced, it's gaining electrons. And these are some just confusing terms you might see: reducing agent or oxidizing agent. But the big ones to remember is which one is reduced and which one is oxidized. So, a couple of questions: is there any oxidation happening? I.e., are things being broken apart? And the answer, are electrons being harvested? And the answer is, of course, yes. Water is being oxidized. We're pulling the electrons off of water, and they're going to go in there. Is there any reduction happening? We see reduction happening all over the place. Chlorophyll is going to be reduced right here. Okay, chlorophyll is also oxidized at one point. NADP is going to be reduced into NADPH. Okay, so all of that is where all that is occurring. Is there any catabolism being happening? Is anything being broken down? Yes, we're breaking down water. Is there any anabolism? Are we building anything? Yeah, we're going to be making ATP from ADP plus phosphates, and we're also going to be making NADPH. So those are all anabolic happenings. Remember, we're coupling these. The breakdown of one molecule is going to release some energy, which is going to be allowed for the building of another molecule. Is there any coupling occurring? Yes, there is. Okay, we're breaking down water. We're bringing energy in. We're using that energy to make ATP, those high energy electrons. All of that is energy coupling. And again, this process, of course, is the light dependent reaction, the light dependent reaction. Okay, so once again, we're using that electron transport chain to create that proton gradient, and then that is a form of potential energy. We're now going to use that potential energy, those protons, we're going to allow those to diffuse, we're going to allow those to diffuse down through ATP synthase. All right, and that is our potential energy. Cytochrome C. Not exactly sure what happened there. I got a different. So here's where I want to get that potential energy is being produced. I'm sorry there. I had a couple of slides that should not have been there. So again, we're going to use that potential energy. We're going to turn that potential energy into kinetic energy by allowing those protons. Again, this is the intermembrane space with a high proton concentration gradient. We're going to allow those to diffuse down through ATP synthase, and in that process, use the kinetic energy of the flow of those protons to drive the anabolic process of building ADP plus a phosphate into ATP. So we've converted the sun's energy into a proton gradient, and now we're going to use that proton gradient, that potential energy, to drive the anabolic process of making ATP. So what do the light reactions produce? They're going to produce energy in the form of ATP, and they're also going to produce electrons and protons that are going to both be used to reduce CO2 to a organic carbon containing compound, i.e. sugar. So remember, CO2 is inorganic because it does not have hydrogen, and we're going to use that CO2 from the atmosphere in order to make a sugar molecule. We're going to use the energy of ATP, which is providing the energy to allow for rearrangement of atoms, and we're going to use the reduction power of NADPH, which is carrying those electrons in order to reduce CO2. So that's pretty much the process right there. And again, the light reaction review. We are taking water in, and in that process, we're going to split water. We're going to energize electrons of water. We are going to use those high energy electrons to create a proton concentration gradient. We're going to use those proton concentration gradient to make ATP. And we're also going to use those high energy electrons to reduce NADP to NADPH. And in that process, we're also going to make some oxygen gas as a waste product. So we now have our ATP and our NADPH produced in the light dependent reactions are now going to be used to drive the reduction and building of CO2 into a sugar molecule in the Calvin cycle. And that is it, guys. I hope this helps.